In this last video, for our genetics videos, we're going to take a look at some of the exceptions to Mendels laws. And these are ones where the traits didn't match the pattern that we saw with some of his experimental results that are out there. And in this example, we have some red flowers and some white flowers. And when these plants were crossed, we got a really startling result with these snapdragons. The first filial generation actually produced pink flowers rather than the white or the... This is, like I said, a startling little thing that's going on there. But what's even more amazing is when you look at this process here and you take your F1s and you cross those with each other, what comes out of there is you actually get those red flowers back, you have pink flowers, and you have your white flowers. And so this process is not really looking at dominant or recessive, but a combination of those two traits in these pink flowers. And if we look at this process known as incomplete dominance, we can take our red flowers with two big R's to represent the red color or the red allele. We have our white flower over here, which has the recessive traits right there. So we're looking at homozygous dominant and homozygous recessive true breeding flowers right here. And when you cross these ones, we can see from our alleles and drop them into our normal Punnett square, that first generation, or the F1 generation that has all the pink flowers, are the hybrids. We have the dominant red and we have the recessive red trait. But for some reason, this red is not completely dominant. It doesn't completely overshadow the white. It's almost like that white allele over here is diluting out some of that red allele. But while these heterozygotes are giving us an intermediate phenotype, again, think about what occurred during that process with our next generation there. Those red and those white colors came back out. In our second example of exceptions to Mendel's laws, we have something that is known as codominance, and this is very different from incomplete dominance. And our characteristic example of this is what's known as the ABO blood groups, or ABO blood typing. And these ones, the alleles are actually found, or the genes or the groups that give us these traits are actually located at multiple alleles or multiple locations on the chromosomes. And the interesting thing about codominance is the effects or the phenotype of these alleles can be seen for each genotype that is out there. So imagine you've got one location on a chromosome that codes for this A, and you have another location on the chromosome that codes for this B phenotype. And what we're really looking at is on the surface of our red blood cells, you can have different types of sugars there, either carbohydrate A, carbohydrate B, or if you're type AB blood, you actually have a mixture. You have both A and B on the surface of your red blood cells. And if you are type O, effectively what you have there is you have two recessive alleles, so you have no carbohydrate A or B on the surface of your red blood cells. And when we look at this process with our Punnett squares, we can see something that's interesting. So if we take a look at this example here we have somebody who has type A blood. They're homozygous, so we indicate this with both capital A's right there to indicate that they have that dominant A allele there. And this individual is type AB. So one copy is that dominant A, and then the other copy is our dominant B trait there. And we take these cells and run them through meiosis again to give us our gametes, and you can see the A alleles are segregated and the A and B alleles are segregated in our gametes over here. We drop them into our Punnett square, and what we see as a result here is those alleles are combined, and so these individuals right here are our type A blood. So half of the kids would be expected to have type A blood, and half of them have type AB blood. So it's really kind of an interesting process when we look at this, and then we can throw in something like our type O blood that is out there where you don't have either one of these dominant traits coming out in that phenotype. And so let's take a look at an example of where we put in a heterozygous type A blood person. They have that dominant A trait on one of their chromosomes, and they have the recessive trait on the other chromosome. we'd go through the process of meiosis to separate those chromosomes or those alleles, and we drop those gametes into our Punnett square to give us our diploid cells here. And if we look at this, what we see is 3 of these individuals are going to have a similar kind of phenotype. So 3 out of our 4 boxes are type A blood, and our 4th box over here, this individual is getting an A allele from one parent and the B allele from the other parent. So they have type AB blood. The third kind of exception that we can look at for Mendels laws is really kind of an interesting one, because in this case you have a single gene on a chromosome or a single allele, and this actually can give you multiple traits coming out of this, or multiple phenotypes. And this is known as pleiotropy. And when we look at this, that single gene, when you have a change in it, you get multiple phenotypes coming out of this process. So the convention sort of has always been one gene gives you one trait, but in this case, one gene is giving you multiple traits or multiple phenotypes. And a very popular example that we see out there is what's known as sickle cell anemia. And in this process, you have a sickle cell allele. And if you are homozygous recessive for this trait, you have both recessive copies on your chromosomes, you're going to end up producing abnormal hemoglobin protein. And when we look at this, it's actually one single base out of 3 billion bases on your chromosomes that can cause this change here in the gene. And when you look at what does this single base change do, it actually results in abnormal protein being made, and you get what are described as very long but flexible chains. Hemoglobin should sort of be a globular molecule where it folds back on itself. The problem with this is that abnormal hemoglobin, when you look at your red blood cells, instead of being these biconcave discs, so think like a plate, they actually end up with this sickle cell shape to them because they are packed full of hemoglobin, and these guys don't make the right form of hemoglobin. And so there is your genotype with the abnormal homozygous recessive trait, and what comes out of this process over here is our phenotype. You see weakness, you see organ damage, pain, and paralysis, and these people also end up with anemia. So they have a problem with the transport of oxygen in their body, and that is more than likely what's causing a lot of these phenotypic problems. They aren't delivering oxygen to all the organs in their body, and if those organs don't have oxygen, remember, you need oxygen for your mitochondria to produce all of that ATP. So these individuals are really running into multiple problems as a result of a single homozygous recessive change that occurred in the alleles that they got from their parents. If we look at our last example with our exceptions to Mendels laws, this is one that is known as polygenic inheritance. And if you take poly, that's many, so we're looking at many genes out there can produce a single phenotype. And a very good example of this is with skin color. You can take 2 individuals, one person with very light skin and one person with very dark skin, and if they are able to have children, what we see here is this sort of intermediate kind of phenotype. So it's kind of what we saw with our incomplete dominance before, with our red and our white mixing together to give us our pink. But here, instead of a single gene encoding this or a single allele, we're actually looking at multiple genes are involved in this process. And if you think about this, each of these ones can end up in our final gametes there. And if you look at your Punnett square for this, you have to take into play every single possible combination of all of these alleles right here. And so you can see that when we get that F1 generation, they have a variety of different alleles that can come down in through meiosis and end up in their gametes. And so you can see all of these different possible combinations coming out of this process with our F2 generation. You can have somebody who comes from 2 parents with medium or dark colored skin out there, and they can actually have a kid who is lacking that dark pigmentation, and you can go to the opposite extreme over here, and the child can actually have even darker pigmentation to their skin. So hopefully you can appreciate that when we look at genetics, there's a big wide world of a whole bunch of different interactions going on. our standard Mendelian genetics, but also we have these other kinds of problems like our gender or sex-linked traits, and then these exceptions to Mendel's rules.